Welcome back, everyone. Our second speaker is Dr. Elizabeth McCarty, who is an assistant professor and forest health specialist in the Warnell School of Forestry and Natural Resources at the University of Georgia. She received her master's in biology from the University of South Alabama and a PhD from University of Tennessee. Elizabeth is a founding member of the Southern Pine Health Research Cooperative, which is tasked with conducting applied research and outreach in the field of forest health in pine plantations for long-term resource sustainability. Elizabeth's work has focused on pine tip moth phenology and management, environmental effects of pesticide use in forests, hemlock woolly Adelgid management, and rare plant conservation. She has developed numerous award-winning outreach resources for pollinator health, conservation, and forests. Welcome, Elizabeth. The floor is yours.
Thank you so much. Let me get this screen shared so everybody can see the slides. All right. Thank you so much for being here today. I'm a forest entomologist, and I work with environmental effects and pest management, as Bodhi said. And so even if you don't work in a forest setting, this topic can be provided as an example for systems you may work in. If you're in ornamentals or agriculture, some of the same risks and benefits of pesticide use we have to think through. So keep that in mind and think through this presentation as how some of this may apply to the setting that you work in if you don't specifically work in forest. Let's see. There we go. So I'll cover the impact on forest health of hemlock woolly Adelgid, different treatment options, and then environmental risks to soil arthropods, water quality, canopy arthropods, and pollinators. And so if you're applying pesticides in almost any setting, these are some risk scenarios that you can think through in your own systems. So what's the resource? First of all, it's eastern hemlock. And this is a tree with a range from Canada all the way south to northern Alabama and northern Georgia. And it occupies a very distinctive ecological niche. This is a slow-growing, long-lived evergreen tree species. We don't have another native tree species that can come in and fill that role. So where we've lost things such as American chestnut, there were other tree species that were deciduous that could come in and in part fill the role of the chestnut. We don't have that in the eastern North America for hemlocks. It provides and conditions the habitat. So hemlock trees can affect the soil, the different plants go around, it shades streams and affects stream temperatures. There's distinctive aquatic insect communities that are associated with hemlock forest. So it really has all these cascading ecological benefits. What happens after HWA has, or hemlock woolly Adelgid has come through and killed all the hemlock trees in a forest is that we're left with open canopy gaps. And in the southern Appalachians, there is a rapid vegetation response. And we really get changes in plant communities, specifically with rhododendron. So if you've ever been through the southern Appalachian forest, you encounter something called a rhododendron hill. And it's called that because they're so awful to get through. Well, those get bigger and thicker and denser. There's changes in soil nutrients, altered wildlife habitat, forest fire intensity, and increased rhododendron density, as I just said. So we're not left with a forest just hemlock is missing. There's all these other effects that happen when we get rid of this species. So I'm not going into a huge amount of detail on hemlock woolly Adelgid, but more of what's our system, what are we left with? If we treat it with insecticides, what are some of the risks that we have to think about, and what do we know about that system? So the goal of hemlock conservation is having a healthy hemlock forest. And because of the role of hemlock in that forest, it's not just about saving a hemlock tree, but it's about having an entire forest system that is conserved. So yes, the trees, but branching out from that, canopy arthropods, soil arthropods, plant communities, water quality, so an entire system. So we're protecting an entire system. We'll use a pesticide for conservation purposes, which is a little bit counterintuitive. We don't typically think about pesticide use being used, for conservation. And so we have this whole system we're trying to preserve, and there's benefits to the system of having hemlock there. There's benefits to conserving the hemlock, but there could be some non-target impacts of the pesticide use. So those are the things that we have to think about when we use a pesticide. There are two different pesticides that are used for hemlock woolly adelgid suppression. They are both neonicotinoids, so that word neonicotinoid may be familiar to you. And there's a lot of information in the media about this class of pesticides. And I would challenge you to be very careful what you read. It's very easy to get on social media and make a comment, to share a post. Not all things that are published in online venues are accurate information. So get your information from sites that end in .gov.edu. State, federal agencies, universities, that's going to be your best place to get information, because there can be information online that is wildly inaccurate. And so you say neonicotinoids, and we have this idea about environmental risk and the public consciousness that may not actually accurately reflect the risk we actually have. So Dinotefuran, its trade name is Safari, moves quickly into the canopy, and it can be effective in about 2 weeks, whereas Imidacloprid moves much more slowly, and it takes about 3 months for it to be effective. Dinotefuran is effective for 1 to about 2 years, whereas Imidacloprid is effective for 5 to 7 or more years. So we're talking about one pesticide application being effective for many years. Dinotefuran is great for quick, short-term HWA reduction, whereas Imidacloprid provides longer-term reduction. Dinotefuran is best for heavy infestation, so it gets up to the canopy quickly and can knock down those populations, where Imidacloprid is best for light to moderate infestations. It takes more time to get the product up to the canopy. If it's a heavy infestation, the tree won't be moving fluid as quickly, and that's how the pesticide moves through the vascular tissue with water as the plant takes up water. In the case of a heavy infestation, Imidacloprid may work, but it's really not the best fit. Dinotefuran is more expensive, whereas Imidacloprid is less expensive. It's off labels. There's lots of different manufacturers who make generic forms of it. So we have two different products that provide hemlock control or HWA control. They're actually very complementary. Both of them are used in ornamental settings. So if you are applying in ornamental areas, Imidacloprid they're both used in forest settings as well, but Imidacloprid is more commonly used because it's effective for a longer time period and the cost is lower. And so most of the non-target work has focused on Imidacloprid. Every time we make a management decision, there's a consequence. The decision to treat a tree with Imidacloprid, the decision to not treat. If it's a pesticide application, if it's whether or not you thin your pine trees, anytime we make a decision to manage the environment, a good decision, bad decision, there are going to be positive consequences and negative consequences. And there's no management decision that we're going to get away from that. It's a trade-off. We're trading off something that we desire in a system for some things that may be negative or may not be negative. And so bear that in mind anytime you apply a pesticide or anytime that you make a decision for the environment, no matter how benign you may think it is, there are trade-offs all the time. So the question comes, we get this idea in the public consciousness that if there's a negative effect, then it is never acceptable. But the fact is that there are negative effects to everything that we do, and we make these trade-off decisions every time we get into a car, every time we let our kids get into a pool. We have to make these trade-offs. So the question really isn't, is there a negative effect, and then walk away, we're done with the decision, but is there a negative effect? How long does that effect last? How large is that effect occurring? And then is that trade-off reasonable for the benefits of the management decision that we're making? So the easiest one to think through in this hemlock setting is that there are canopy arthropods that live in hemlock canopies. There are over 400 arthropods that are associated with hemlock trees. So it's providing that habitat. And if we use a systemic insecticide that moves up through the vascular tissue of the canopy and targets a fluid-feeding insect in the canopy, then it's not very realistic to think that no other canopy insects would be effective. And that's not a reasonable expectation. But, how much does that effect occur? How long does it last? And then what's the consequence of not treating? In the case of hemlock, that's pretty easy. The tree is going to die. And then those canopy arthropods will no longer have a habitat. So this is, again, the easiest trade-off scenario to walk through in a hemlock setting. And this is much more simple than many of the trade-offs that we have to think through in our own jobs. So it would be great if we had this magic equation or balance, and we could put the consequences of hemlock loss on one side and the consequences of hemlock management on the other side, and see what the balance is. Are we getting more benefit from treating the tree, or are we getting more benefit from just letting it die? Are the non-targets just not worth it? And the fact is that we don't have this perfect equation where we can get it to an exact number. So that means that we have to gather information and then think through these things, have conversations, and really try to understand the whole situation. And you may notice that a couple of things are the same. So some species that may be affected by hemlock loss may also be affected by using a pesticide to manage hemlock. So those are things that we have to think through carefully. This is one of my favorite quotes to share when we talk about pesticide issues. For every complex problem, there is an answer that is clear, simple, and wrong. And that is very true of these settings. These are very complex issues. It's messy. There's lots of things to consider. And it's a conversation and not a one-liner or a meme or a quick Facebook post that gets thrown up. We have to really think about these issues and consider them carefully. And one good thing is Mickey had all these things that he discussed with the pesticide label and the restrictions of the pesticide label. And those restrictions are for safe pesticide application. They provide a safety fence for us to use a pesticide and minimize the chance of having effects that are not desirable. And so part of this question, it can be answered by correctly following a label, and that can avoid a lot of potential problems. So let's walk through this risk scenario. Imidacloprid is applied to the soil around the base of a hemlock tree. Those applications occur within 2 feet of the trunk of the tree. The expectation is that it will move up into the tree canopy to kill HWA and possibly have some impacts on non-target canopy arthropods. And we know that may happen. And so are those impacts reasonable, and what's the trade-off? What else may happen? It could affect soil arthropods right at the base of the hemlock tree or extending further out. How far out does that extend? Is it reasonable to think that I would pour a pesticide around a tree, and the soil that I pour that pesticide directly on, it would not potentially kill some calimbolas and ants? That's not a very realistic consideration. If I'm going to pour it directly on the soil, I may kill some things. Again, the question is how long and how far does that extend away from the application site? There could be movement through the water, through the soil into the water. We could get imidacloprid residues in the water, which we do. And then the question is, how long does that persist? And do we see any biological impacts of that? And so the first 3 areas, the canopy, the soil, and the water, I'm going to present data that has already been published, some of it that I've done and some of it by colleagues, to help answer those questions. And then lastly, and this is a study that is ongoing in my lab, it can move through the soil, be taken up by non-target plants that are growing close to the hemlock tree, be incorporated in those plant tissues, and potentially affect things like pollinators. So note that the picture of the plants, these things look like they're 8 feet tall. In reality, we're talking about plants that are less than a foot tall and are growing right around the base of that hemlock tree. So let's put that risk into perspective. So we're going to walk through the different areas of risk in these forest settings. But remember, if you're applying in a yard or a park or something like that, these are similar risks that may occur. Or if you're applying in other systems, that's a great thing to bring up during the question and answer session and how we think about the risk in different settings. So first, soil arthropods, and I show a picture of a calimbola first, a couple of calimbolas, a mite, and then a soil profile. Calimbola are really good organisms to assess for non-target impacts. They're relatively sensitive, and they've been assessed in many different agricultural and forest settings. And one thing is important to remember, use of pesticide in forest is very different than use of pesticides in agricultural settings. And that being said, the environmental benefits and our tolerance for non-targets is probably lower in forest settings than in an agricultural field. There is some vertical movement in the soil. So this would be down the soil column. So we know that there is some vertical movement, and that is from a study by Knaup et al., 2012. There is, at the time that this study was published, there was limited horizontal movement. So that is side to side in the soil profile, away from the application site. And I have question marks there because in a study that I'm doing with the plants, the non-target, I'm seeing higher concentrations move away in the soil, away from the application site. But the difference is that the Knaup study used lysimeters, and that's where they're pulling water up from the soil pore spaces and looking at the concentrations of insecticides in that water that's in between the soil spaces. Where I'm using bulk soil concentration, so just what's in the whole mass of soil. And we now know that the water content in soil has a lot to do with how toxic imidacloprid is to organisms. So just because it's in the soil does not necessarily mean that all of it is biologically available at that time. But what they did find is that there was no population difference between treated and untreated trees. So when they looked at the arthropods living in the soil, things like mites and columbella, treated and untreated trees didn't see a difference. And they looked right at the area of application and extending out toward the drip line of the tree, so where the branches end. There was a weak negative correlation between imidacloprid concentrations and populations at one site deeper into the soil, but the correlation was very weak. So I wouldn't hang too much on that. But the big take home was that there wasn't a difference in treated and untreated areas overall. And then there was no relationship occurring at the soil surface for imidacloprid concentrations and the arthropods. And that's where we're expecting those higher concentrations to be right at the surface where the imidacloprid was applied. So that's what we know about soil arthropods. And I'll cover a little bit on ants later on in the presentation. So let's step away from the tree a little bit and think about surface waters. Are we seeing effects in the water as the imidacloprid moves through the soil away from the tree? So imidacloprid will bind tightly to the soil, but as water comes and flushes through the system through rainfall, it will detach from the soil and move at lower concentrations. As it moves away from the application site, the biggest degradation is occurring through bacterial degradation, breaking that compound down. And then it's moving further away from the tree. It's diluting into larger areas of soil, and then getting into the water column. And it does get into stream waters at very low concentrations. And I show this map. This is for the study that I'll talk about. This is my doctoral project at Great Smoky Mountains National Park. And all of the spots indicate where samples were collected throughout the park. So very wide geographical area. I'll cover the aquatic macroinvertebrates. What we did is there was baseline data by the Park Service, the species list of what was occurring in these areas before imidacloprid had ever been used in the park. And we had downstream areas, and these would be where our treated samples. And then the area that is indicated by the red box is where imidacloprid soil treatments had occurred in trees that were in that riparian corridor. And then an upstream sample was collected as a control. So we've got our treated, the downstream area, upstream as a control, and then the baseline data as an additional control. And then our areas where the treatments occurred, soil treatments and riparian corridor. And we looked at sensitive aquatic insects, the mayflies, stoneflies, and caddisflies. So what we did is looked at all of the downstream, those would be 9 stream pairs, and all of the upstream sites to assess this question. We had already done water samples in the area, and out of the 9 streams that we sampled, 6 of them did have imidacloprid. With the exception of 1 stream, all the concentrations were below 100 parts per trillion. So again, very low concentrations in the streams. And what we found is we looked at different community indices. What this means is things like abundance. How many different insects did we find at each site? Richness. How many different species were at each site? Dominance and evenness tell us, do we have samples that are dominated by one organism, or is it a good mix of things? And that's important because if you have an area that's impacted, It may be that one tolerant species, one species that does not really care if it's polluted or not, will end up dominating the system as the species that are more sensitive end up dying out. And so having something that was really dominated by one tolerant organism would say, oh, wait a minute, I think we have a problem. But what we found is that there were no significant differences between upstream, downstream, and the baseline data that we had. So there were 552 insects per stream site in our samples in the downstream location and 445 in the upstream location. If the downstream number was a lot lower, that would indicate, hey, we might have a problem here. On average, there were 22.9 species collected in a downstream location and 23.7 in an upstream location. So not a significant difference. If this was really low, it would say, wait a minute, we might have a problem. I'm looking at all of the other measurements that we collected to tell us what's going on with these communities. We didn't see significant differences. Mean tolerance value is one that I like to point out. This is one that's used in water quality testing a lot. And the tolerance value is ranked from 0 to 10. So 0 is something that is very, very sensitive. And if the environmental conditions are poor, that organism is not going to live. 10 is very tolerant. So these would be things that we find in highly polluted urban streams, things that are downstream from major pollution sites, and only these very tolerant things would survive. So there was no difference, and the mean tolerance value for these numbers were very low, indicating that the communities that are occurring in these sites are very sensitive. If there was a problem, then we would see this number being much higher, indicating that we were losing our more sensitive organisms and keeping a lot of the tolerant ones. So just in an overall, what's happening upstream, what's happening downstream, we're not seeing a negative effect. All right, but let's take a step closer. Let's look at more than just what's living here, but how are these communities functioning? So we have different feeding groups: collector filterers, generalists, predators, scrapers, and shredders. This tells us how are the different organisms feeding. And then life habits: burrowers, are they burrowing down to the sand? Are they clinging onto like sticks and branches? Are they sprawling along the bottom? And this tells us how are the animals behaving in the stream. So there's two different ways to look at how those communities are functioning. And then we have different ways that we can measure them: the abundance, how many different organisms did we find, the richness, how many different species we found, and the proportion, what percentage of the community is made up by these different types of organisms. And instead of showing you a bunch of different numbers, I'm just going to color code it. No difference. Downstream is better. Downstream is worse. And so I would expect to find some no difference, maybe a couple spots where we'd have some better or worse upstream or downstream. But if I found a lot of red, then that would give me concern. That would say, wait, we're getting a lot of different metrics here where our downstream looks less healthy than our upstream. And so what we found with this is that there's no difference, not only in all of the diversity metrics that I just talked about on the last slide, but also in how the different groups feed and how they behave in the stream. And that's comparing upstream and downstream. We also had our baseline comparisons, and these were, I'm only going to compare richness because we just had species lists from different park samples. And there's one incidence where there's a red and we have a blue and most of it's green. So this is like a little ding in one direction. That's a little ding in the good direction. So nothing to write home about. I think looking at all of these different assessments, there's a lot of no difference between upstream and downstream, indicating that although we do get a little bit of pesticide in the water, it's not happening to a degree that's causing problems for our biological communities. Imidacloprid is more toxic to invertebrates. So this whole class of, because a question I get a lot at the end of this is, what about fish? Are we worried about fish? This class of insecticide was developed because it had a low toxicity to vertebrates. So it's much more toxic to invertebrates and much less toxic to vertebrates. So the concern with fish, it takes a lot of this pesticide to kill vertebrates. So at the levels that we're finding, I'm not concerned about fish getting a toxic dose of this pesticide. The next question would be a food web effect. are we killing a bunch of the invertebrates in the stream that would cause issues for the fish? Are they losing a lot of their food? And based on this study and others, the answer to that question is no. We don't anticipate seeing an issue with fish because their prey is being compromised. There was another study that was done at the University of Georgia where imidacloprid treatments were applied in a similar way in the riparian areas in 4 different streams. And they collected water and aquatic insect samples for 2 years after treatments were applied. They only found 1 sample that was positive for imidacloprid about 2 years after treatment. The limit of detection, how low they could go in concentrations, was about 600 parts per trillion, where mine was about 15. So it may be that it was present and they weren't able to detect it. But either way, they did not find any negative effects to the aquatic insect communities. All of the streams had excellent water quality using aquatic insect metrics. So we've got 2 different studies in forest looking at this pesticide, seeing not being able to pick up on negative effects when they're being applied according to the label. All right, let's move next to canopy arthropods. And if we're going to see an issue, this would be where I would expect it, because our target organism is a canopy arthropod. So it would be reasonable to expect some effects to canopy arthropods. So 3 different studies I'll mention. 2 studies are a shorter term study, 1 to 2 years after treatment. This was done by Carla Dilling, who is a colleague and very good friend, excellent scientist. She did a species level assessment, so very detailed study. Falcon and DeWald in 2010 did an order level assessment, so not as in depth on the identifications, but theirs was part of a wildlife study, and they were answering that food web question. Are we seeing effects in the food web? And I'll present bird data as well from that study. So Dilling et al. Found some negative effects initially on abundance, the number of insects in the canopy, especially for soil drench. It was not a complete wipeout. If we saw a complete wipeout, that's where we would say, wait a minute. There was a decrease in abundance, but not a catastrophic decrease. 33 species were directly affected, moths and bark lice. So this is a picture of a bark lice. And then moths, and all the moth species that were affected are ones that pupate in the soil close to the base of the tree. So it may not necessarily be that they were affected by feeding in the canopy, but they were affected by pupating in the soil and getting that dose from the soil. So yes, some initial effects. So when we think about that tipping scale, we're never going to get a situation where we don't see any effects. We have some effects. How serious are they? Are they catastrophic? No, but they are there. This is observed 1 to 2 years after treatment. And we're talking about treatments that last 5 to 7 years. The next study, Falcon and DeWald did an order level assessment. So only going down to insect order, and that's more of looking at biomass. How many insects, what's the abundance of insects on this canopy that birds are going to be feeding on? And there was no difference in total abundance or the abundance of even herbivore insects in the canopies. They did find some relative abundance, so that would be the proportion of these different herbivores. And so you're seeing other insects groups become more predominant. But again, no catastrophic, some significant impacts, but no catastrophic impacts. So that's good to know. We do get some initial impacts. How long does that last? All right. Then comes to the next study, Kung et al. And this study was conducted further north, where the first 2 studies were conducted in Tennessee. This was up in Connecticut. And they looked at hemlock trees, treated and untreated, similar types of metrics with canopy arthropods, and they found no negative effects 3 years after treatment. So we see in one study, initial 1 to 2 year effects. In this study at 3 years, they were not seeing effects in richness or abundance, these metrics that we look at to see what's going on. 9 years after treatment, they actually had higher species richness in hemlock canopies. Okay, so what does that mean? 9 years, these trees had gone and they had not been treated. They still had healthy hemlock canopies, but at that point, the untreated trees, their canopies were declining. They were starting to really feel the effects. See, further north, they get winter kill for HWA. So a tree can hang on for 10 or more years and be infested, where further south, hemlock trees are killed in 2 to 3 years because we don't get winter kill for the HWA populations. But what they found was that the hemlock trees that were treated had higher species of arthropods than the trees that had not been treated, that were still alive, but had really unhealthy hemlock canopies. And then let's look at a vertebrate because it ties in with some of the canopy arthropods. This is a study that was conducted in Tennessee. They looked at 3 different migratory bird species that are associated with hemlock canopies. And they assessed the number of bird territories and the density of those territories in 6 treated and 6 untreated sites. And they found that there were just as many bird territories in one treated as compared to untreated. And they're looking at territory because that's the best way to measure the birds. Instead of trying to count and uncount birds, they look at the different territories. So there's no effect on the density of these territories. They also looked at the hemlock foliage mass. So in untreated areas, hemlock foliage was starting to decline, and there was a lot less foliage on those hemlock trees, where in the treated areas, the foliage was still there. There was more foliage mass. And they found that whenever there was more foliage mass, that was related to how dense these territories were. So the birds were benefiting by hemlock being present in the system. And then there was also a negative relationship between dead woody material. So as there was more dead wood on the hemlock trees, there was less bird territory in those areas. So that was being a negative effect of hemlock mortality and a positive effect for the hemlock being there. So lastly, I'll talk about pollinator risks. And this is highlighting a study that I have going on in North Georgia. So I've been at Tennessee and I had been working in hemlock canopies with insecticide and how effective it was. And I was also working with streams. And we started asking this question about how much can this move in the soil? We know it's moving because it can be in the streams. How does it move away from the tree? How concentrated is it? And do we see effects in non-target plants where it would be affecting insects? And I was approached by Forest Service ecologists who were concerned about persistent Trillium. This is an endangered plant that is found in the Georgia-South Carolina border. There are 6 isolated populations of persistent Trillium. It is not endangered because of hemlock decline. It's endangered because these populations are small and isolated. But it is further threatened by hemlock decline because these are only found in mixed hardwood hemlock groves. So we don't find these plants away from hemlock trees. So they need that habitat. And it needs the habitat created by the hemlock, pollination, and ant-mediated seed dispersal. So there are 2 different groups of insect associates for this Trillium, the pollinators and the ants. And what the ants do is that they take the seeds and move them away from the Trillium, and that way the seeds are dispersed further in the environment, and that population can grow away from the parent plant. And then at the time that we started this study, we didn't know who was pollinating the Trillium plants. Different Trillium plants can be pollinated, different species can be pollinated by entirely different orders of insects. One may be pollinated by a beetle, one may be pollinated by a fly. So what's the risk in this system? This is messy. We have a habitat that's threatened by HWA. The pollinators and the seed dispersers could be threatened by that too, because once hemlock's out of the habitat, we start to have these cascading ecological effects. We're using an insecticide, and either of these insect associates could be impacted by the insecticide. And let's add in here that I'll mention later, there's an invasive ant at these sites called the Asian needle ant that can compete with the seed disperser and further threaten this species. So this is not an easy system. There's a lot going on. This project had 2 objectives: to evaluate the risk to persistent Trillium pollinators and to seed dispersing ants. So there are 2 different sites. There are 6 different subpopulations of this Trillium. I would love to have 4 or 5 sites for this study, but we're limited because we're dealing with an endangered plant, and every site's not really conducive to the research. And we don't want to do research on every site. We want to be really careful with these populations. So there are 2 sites. 15 trees are treated at each site. And we used 3 different treatments to see which one would be most protective or more protective at all of these insect associates. The soil drench, that's where we pour soil on the base, around the base of the tree and the soil. Soil injectors, where the pesticide is injected a couple inches below the soil surface. And then bark spray, where the insecticide is sprayed directly on the bark of the tree. It moves through the bark and then is translocated from the vascular tissue up to the canopy of the tree. Bark spray is interesting. Most research has been on soil drench and soil injection. And then about 6 or 7 years ago, we were hearing a lot of people talking about bark spray and how it was more environmentally friendly because the applications were being made to the bark. And in theory, nothing's getting on the soil and that insecticide can't move away from the tree. But nobody had actually researched it. The idea of it's good, but we don't actually know how accurate that is. Some states further north will not allow soil treatments. So for instance, in New York, they can only use bark spray. They're not able to do soil drench and soil injections. So that's something to bear in mind. All right, so let's lay this study out and walk through a little bit more details on it. 15 trees at each site, 5 trees treated with soil drench, 5 with soil injection, and 5 with bark spray. And then under each tree, and so we've got the representation of the herbaceous plants down here. And this is a little bit more scaled accurately. We collected 2 soil samples, 2 foliage samples, a goal of 2 floral samples, and 1 leaf litter sample. The soil, foliage, and flower samples were used to see how much insecticide residue is moving away from that tree into different types of environmental tissues. And the leaf litter was sampled to assess the ant communities. The leaf litter samples were collected right with the square meter right at the base of the tree. And then the soil samples were collected within 2 feet of the tree. One interesting point with the floral samples, we had a goal of collecting 2 samples per each tree and only persistent Trillium. When we got out there, even despite numerous recons, there's just not enough density of this plant to collect 2 flowers under each tree. So on average, we only collected 1 flower under each tree. All samples are collected from the downslope side. This would be the area that would have the highest risk of getting pesticide. And so we've got this picture with all these flowers under the tree, but that's a little bit of an exaggeration of what we actually find in the field. So in March, before the treatments were applied, we collected soil, foliage, and leaf litter samples. And then those are controls. I applied the insecticide. And then in April, we get all the samples plus floral samples. And then in August, 4 months later, we collect samples again. So there was pre-treatment. The insecticide was applied. 3 weeks later, we collect samples. And so this is really going to be where we think we're going to get the highest concentrations in the flowers, because at this time, the flowers are developing and it's pulling in water from the ground and putting it into flower development. It's a lot of resources go into those flowers. So 3 weeks later, that's when we're expecting to get a lot more insecticide. 4 months later, 1 year later, a year and a half later. And then we were supposed to collect samples in 2020, but we could not safely do that with COVID. So we'll pick this back up again in April. So let's think about the landscape. All right, we're looking at the trees. So each circle indicates a tree, and 6 feet circles have been drawn around each tree. And so these are more like little contamination islands. So if we see an effect, it's not necessarily occurring in this entire polygon and this entire site. It's occurring in this little island around the tree. And so if we add up all these islands and look at the total site, we're talking about at most about 5% of that area. And then these 2 to 5 acres, depending on the site, is then embedded in a larger landscape. So one question I've been asked is, did you sample the insects, the pollinators flying through this forest? And that really won't give us the answer that we need, because pollinators are going to be very mobile and they'll be flying in and out all over the place and in and out of these little contamination islands. So sampling pollinators may not really give us the answer that we want. it's not really answering the question because of how mobile they are and because of how the contamination is occurring in little islands around the tree. But we looked at soil concentrations right around 24 inches of the tree. So this is right where the insecticide is being applied. Foliage within about 6 feet of the tree. And we had to do that because we didn't get enough plants even around the tree to collect within 2 feet of the tree. And then flowers, we had to move out to 74 inches. So we extended out more than expected to be able to collect the samples that we needed. And that does speak to risk in the system. Again, the goal was to collect as much persistent Trillium as possible, and about 50% of our samples are persistent Trillium, and the rest of them are made from other herbaceous plants. So first, let's look at imidacloprid concentrations in the soil. All right, we start with March. Don't have much in there, a little bit of residue from treatments years before. And then right after treatment, it pops up to about 8,000 parts per billion. And that is high concentration. And if an insect were to eat 8,000 parts per billion, that would be a concentration high enough to kill them. But we know from other research that just because that concentration's in the bulk soil doesn't mean that it's all biologically available. It has a lot to do with moisture. So then we're going to rely on looking at the Arthropod data that I'll share later on. And that concentration stays high for about a year and a half, and then it decreases down significantly in August 2019. And so we'll see what the numbers do in 2020. That'll help us to get an idea of that risk. What is it initially, and then how long does those concentrations persist in the system compared to the entire lifetime, 5 to 7 years of a hemlock treatment. Also in August, I took samples that were 10 feet away from the trunk on the downslope side. And those concentrations, while they were 5,000 parts per billion in the soil, they were 38 10 feet away from the tree. And then 23 the next year. So as we move away from the tree, as I said, it's diluting, it's getting degraded and getting diluted in the system. So moving away from the tree, the risk goes down. All right, imidacloprid concentration in foliage. So imidacloprid concentration in parts per billion. And note that the concentrations are much lower in the foliage than they were on the soil. In April, we get a significant jump up to 600 parts per billion. And then it goes even higher in August. And so we see this trend across the years where it's lower in April and then increases, maybe not significantly, but numerically it's higher in August. Why does that happen? The tree starts growing and the plants start growing in the spring. And over the course of the season, they continue to grow, bring up water, and incorporate the pesticide into their tissues. And then most of these plants are going to die in the winter and then start regrowing again in the spring and incorporating the insecticide through the course of the growing season. And so I would expect to see this trend of lower concentrations earlier in the year and higher concentrations later to occur in 2021, but even at lower concentrations. Now, I will note there was no significant difference between any of the insecticide treatment measures. So bark spray, soil drench, and soil injection. No difference in the soil, no difference in the foliage concentrations, and no difference in the flowers. So concentrations in the soil of a bark spray are just as high as the soil treatments. So that does provide information on, are they more environmentally friendly? And I really don't like that term, but are we seeing that better environmental profile because it was applied to the trunk? And this study suggests not. That's not a bad bit of knowledge. It's not good, it's not bad, it just is. But I did take this picture when it happened. I use a low flow sprayer and I make my applications very carefully. And I noted that occasionally the spray would hit the bark at the wrong angle and we would get backsplash onto non-target vegetation. And I'm a very careful applicator. A contract applicator trying to go fast may not be as careful. And so we would get this back spray onto the plants. And in fact, our highest foliage and floral concentrations from persistent Trillium, and these are individual data points. So that's not overall, but just if it splashes off the trunk and lands on the foliage, it can result in higher concentrations in those tissues. And lastly, the risk to pollinators, 500 parts per billion, 3 weeks after treatment. And so that is high. That's higher than I would like for sure. And then it goes down significantly. All right, but let's put these numbers in perspective. What do those concentrations mean? Right, the EPA no effect concentration for honeybee is 25 parts per billion. So what that means is that is the concentration at which no effect is observed in bees. So they don't see a problem in bees at this concentration. The pollinator for this is a fly, a very small little dance fly. It could be much more sensitive than honeybees. We don't know that right now, but what we do know is at least by the second year, concentrations are getting down into a range that is more acceptable. So higher initial concentrations at 1 year and then lower. Is that good? Is it bad? It just is. And then we're able to start making decisions based on that information. Remember, this is not every flower in the forest. This is just the flowers that are occurring very close to the base of these hemlock trees. And lastly, the ants. So we're getting concentrations in the soil. All right, let's take that fact. They are in the soil. Are we seeing a negative effect because of those concentrations? All right, first of all, we encountered a surprise, and that was the invasive Asian needle ant. And the invasive ant has a range from northern Florida to Connecticut. It's around. It probably hasn't been studied as much as it should have. It doesn't require disturbed habitats. So fire ants and a lot of other invasive things, they like areas of disturbance. Asian needle ant doesn't care, which means that it can penetrate deeper into undisturbed areas like forest. And it competes with native ants by barging in on their habitat and eating them. It predominantly eats termites, but they can also eat native ants. Overall, we found 338 ants among all the samples, 13 different species, 3 of which were seed dispersers, 12 native species, and this 1 exotic. So overall, there were 0 to 4 ant species per sample and 0 to 14 ants per sample. Between the 2 sites, 56% of the ants were the invasive ant. So this is not good news, and this was not anything that we were expecting to see. 26% of the ants were non-seed dispersers, and 18% were seed dispersers. So is this an amount we would typically see in a forest? I'm not sure at this point, and I will be digging into other studies to find out, are we seeing much lower levels than we want to see? And the driving factor of that is likely going to be the invasive ant. And so we have the seed dispersers. These are the guys that we want to be there. Should they be 100%? No, but I'm not really sure what level is acceptable. So first of all, the number of ant species or their richness. The treatment method did not affect the number of ant species, and the timeline did not affect. So comparing before treatments were applied, 3 weeks later, 4 months later, we had to drop these samples due to a technical issue in the field. Then a year and a half later, we're not seeing an effect of these pesticide application to ants. And we did place our sample locations right at the base of the tree at overlapping where we poured the pesticide. So they appear to not be affected. And then the number of ants per sample. And again, the same trend among the different treatment types, and also over time, not seeing an effect here. So that is encouraging. All of the other arthropods in the samples are being identified by the Natural Heritage Museum at Clemson University, and we're going to incorporate that in to see if any other groups are being affected. So walking through this situation, the habitat is threatened by HWA, as is the seed dispersers and the pollinators. Asian needle ant has popped up in here, and so we have a messy system. So questions that we have to ask are, what proportion of the Trillium, as well as all of the other herbaceous plants, are exposed to the insecticide treatment? We're talking about 5% of that forested area at the most, so a low level. How long do the insecticide concentrations persist? There are trade-offs, right? We're getting pesticide in the soil. We expect that. We're getting pesticide movement into plants and flowers. We expect that. So what are the trade-offs between a high initial concentration and lower concentrations later? And can we do anything in the field to reduce the risk? For example, if we treat in the fall, does that reduce the concentration that occurs or reduce the risk? Or if we treat after the plants have bloomed? So it's not like absolutely not this, we're going to take this 500 parts per billion out of context. We have to embed that fact into the system and make decisions based on that fact placed in context. And then can the public think through these trade-offs? If it's the National Park Service or state forestry agency, then there is a public that they have to answer to. And so that's where science communication comes in and framing the results in a way that is non-biased and well-rounded. So this is wrapping up my talk. I understand that you may not work in forests or in other systems, but these different risk scenarios may play out in your system in different ways. And so even if it's a different system, we can take that same walk through thinking about risk, thinking about facts, and how we put them in context to the setting. And so I'll be glad to take any questions or talk through this risk if anybody would like to put it in the Q&A.
Thank you, Elizabeth. This was wonderful. And I really love the way you gave us this balanced approach so that we can think about how complex a system is and how there are really no easy answers and how we should really try to get as much information from as many reputable sources as possible before we make our decisions and really stay current with research as we know. So there are a couple of questions that came in. Would you like me to read them or would you like to do it yourself?
I can read them, and I'm going to stop my share because there is one question that I will end up pulling up a reference to look for. So why don't you read them, and I will look up something for the woolly hackberry aphid.
Okay. While you're looking at the woolly aphid, Mark is asking a question. Do you have any experience using Cortect on hemlock?
Yes, Cortect is a nice option. It is great for people that, especially when I work with Great Smoky Mountains National Park, has largely gone over to Cortect because for practical reasons, it's easier to take Cortect in the field. It's lighter to carry, and then the applicators are not having to carry in pre-mixed pesticide or they would have to go down and find a water source. So for areas where they are hiking in numerous miles to do these treatments, Cortect is a good option. I did some preliminary work when I was there looking at concentrations 5 years after treatment, and it didn't have the replicability for a publishable study, but it did give them confidence that it would last numerous years after treatment, and it was a good method. If you will Google optimized dosage and then UGA, you should be able to, or I'll put it up in the chat in just a minute. There's optimized dosage recommendations for Cortect that we've developed that can also be used if you're interested, and that would let you use less Cortect and provide good treatment for hemlock trees. So we can reduce the amount of pesticide even lower than label limits, and that is research-based information.
Great, thank you. Jeremiah has asked, has there been any comparison between predatory beetles and pesticides for treatment of HBA?
Yes, there has been a study. It was published in 2015. And so you would not want to treat a tree and then immediately put beetles on it. There needs to be some time. The U.S. Forest Service this year came out with their recommendations for integrating chemical control and biocontrol. That is a very specific recommendation. I peer reviewed the information for the Forest Service. It's an excellent resource, but biocontrol is the long game. And there are outfits that will sell you biocontrol and tell you that it will effectively control HWA on the trees that you release the biocontrol on. And that is not accurate. And they are scamming you for money. And I can name the operators, but I'll be professional and not. If you find somebody who is selling you beetles to put on your individual trees in your yard, do not buy them. If your goal is to save the trees on a specific area of land, then you need to use a pesticide. Beetles are more appropriate for federal and state agencies or people working with those agencies, knowing that the trees that you release the beetle on will largely just be part of building up those beetle populations in the environment, and they may not necessarily and are very unlikely to survive. Does that help answer that question?
I think it answered it for me. We have only about a minute more to go.
All right. For aphids, both Imidacloprid and Dinotefuran are labeled, and you can find that in the Georgia Pest Management Handbook. in the tree section on the commercial edition. And so if you have any pesticide questions, the Georgia Pest Management Handbook is the place to go. That section for forestry and Christmas trees was updated, or at least the forestry trees was updated and rewritten by myself and Will Hudson in 2012. It also includes tables on pesticides and which ones are labeled for different pests. That's a great resource. And I
Thank you.
Chat.
The new pest management handbooks are expected to be delivered around the 14th of February, and we'll be getting them ready for sale at that point and start providing them to people.
And I'm just putting the web link for that chapter for forest and tree pest in the chat.
Great. It certainly sounds like you have a lot to talk to us, maybe in a future webinar. Maybe we'll get you back next year and you can share with us results from this year, and maybe you can talk about pine tip moth then, huh?
I'd love to come talk about pine tip moth.
I'm sure you do.
I
have a question that has very little to do with what we've talked about. I have been watching your slides, and there's this one slide that has a little green striped little worm thing that I was wondering what it is.
That is a hemlock looper. And that's important. It is a chewing pest, and the hemlocks in the eastern U.S. have co-evolved with that pest. So all of their defensive compounds come out when that pest is occurring. All of the other hemlock trees in the world evolved with Adelgid, the sucking pest, and all of their defensive compounds occur when that sucking pest is active. And so when you put that sucking pest into eastern U.S., the trees are not evolved to produce defensive compounds at the right times for them.
That just opened a whole bunch of possibilities for more research for you and more questions for me. So thank you so much. Thank you, Mickey, as well. We really appreciate you starting us off to a great start for the GT BOP 2020. Thank you again for joining us, and we hope to see you in future webinars. Thank you.
Thank you.